All right, thank you everyone for coming. Please find a seat, and we're going to get started. I'd like to welcome you to the first annual Gottfried Schmier Memorial Lecture. There are formal introductions, but as the interim chair of the department, I get to put in my own 2 cents. I wanted to say 2 things, and the first thing was that Gottfried was my friend. He was a close friend, and I spent a lot of time with him towards the end of his life. And I really miss him, and I really am happy that we're able to honor his memory in this way. The second thing I wanted to tell you is you're going to hear an introduction by another friend of Gottfried's who really understands a lot about his background in coagulation research and clinical practice, and then you're going to hear someone who has a connection to Gottfried in his second career in tropical medicine. My connection to him, I'm not a coag person or an infectious disease person, but I'm a chemist, and I wanted to just let you have just the tiniest bit of knowledge about what he did as a chemist. This is a paper from 1971 in JBC. He had come here in '67 as a postdoc to work with Earl Davey. We have Earl Davey's son with us here today. In the present communication, he describes a relatively simple method for the purification of milligram quantities of bovine factor VIII. He later purified also human factor VIII. But this is his relatively simple method. I read to you from the abstract. The purification procedure involves barium sulfate, kaolin, and bentonite absorption to remove contaminants, ethanol, polyethylene glycol, and beta-alanine fractionation, calcium citrate, cellulose chromatography, concanavalin A, precipitation, and agarose gel filtration. That's the relatively simple procedure that Gottfried invented. He did biochemistry back in the day when it was real badass stuff. And I really admired him for that, and it was great comparing notes. But to get on with the show, I'd like to introduce Dr. Wayne Chandler, who will be giving the introduction.
It's my distinct pleasure to give introduction to our speaker for the first honorary Gottfried Schmier lecture. You've heard a little bit. He came here to the United States to do research in the 60s, and then he joined the Department of Lab Medicine because he had an interest in coagulation, and so he essentially took over and set up our first coagulation division and ran that for years. I first met Gottfried in 1982. Back in the days, there were no telephones. When we wrote in the chart, we used a hammer and chisel, et cetera, et cetera. So, and this is kind of Gottfried around there, and he was really an interesting guy to work with. We had lots of good teachers in lab medicine, but Gottfried definitely stood out. As you can see, if you look in NW 150, I think they still have the list of who gets the resident award, and you will see the resident teaching award on there for Gottfried multiple times. So in '82, I met him, and it was really fun. It was a good introduction. He would want to know all about the patient, history, physical, what was going on, what coag results did you have, and then you wanted to put the whole picture of the patient together. He wanted to know if you'd seen him and read the chart. If we had something to add, we would talk to the physicians, occasionally write a note in the chart. So it was really quite an introduction to sort of how to be an overall, clinical pathologist. And then it was kind of fun because then after you'd talk to him about the cases for the morning, then he would say, well, we should go to the lab, and kind of see how things are going, meet with the techs and see what's happening in the lab. And of course, there was a classic line for that. He said, "We are now going into the trenches of warfare." So that was a classic Gottfried. He has a bunch of those. And so I had a thoroughly enjoyable time with him, learning from him and sort of being mentored by him. And I knew him for years. I continued to know him even, weeks before he finally passed. I was still sitting with him over at Children's. He would come over every Thursday for grand rounds at Children's, and we would sit together and sort of say, how are the kids doing, and this, that, and the other. My kids are smaller than his kids, but mine are getting bigger. So it was, it was fun. This is Gottfried at my retirement dinner, and he was giving a good retirement from the University of Washington. I sort of wish it was retirement now, but not there quite yet. So, he was active to the end, and you're going to hear about what retirement was for Gottfried. Well, retirement was learning about tropical medicine, and I won't go into it detail, but going down the Amazon, and I can still remember we would talk about sort of his experiences in the leprosy clinic at Harborview. And one more anecdote, and that is that, of course, he wasn't just looking at the tropical medicine side of the individual. he was doing a physical exam and checking them out, and he had talked to one person, or he had examined one person and listened to their heart carefully and take a history from them, and he came out and he said, and this patient had aortic stenosis, and I told the other physicians, and I said, this is what we should do for him. And they said, well, you can't diagnose aortic stenosis that way, and so they sent him to cardiology clinic, and he got echoes, and he got this, and a month later he came back and he said, well, what do you think? And I said, well, he has aortic stenosis, I guess, and we're going to do kind of what you said. And so they kind of, and he said, I think nowadays we fool around a little too much. So that was Gottfried. He was a great clinician, good laboratorian, infectious disease, parasitology, internal medicine, great teacher. I tried to sort of model myself after him, dedicated researcher, and as others said, a great friend. And now I'll give a brief introduction to our first speaker for this, Dr. Sean Murphy, BS in microbiology with honors from Iowa State, got his MD and PhD from Northwestern University, and then moved up from those to the distinguished residency at the University of Washington, and then was appointed assistant and now associate professor at that same distinguished department. So without further ado, Dr. Murphy.
Thank you very much. Thank you to Peter Schmier for traveling here all the way from Florida through, if some of you read the news last night, horrible delays in O'Hare. So thank you very much for being here. I'm honored to give this lecture today, and I'm pleased that my whole lab group is here in the front row to join me. I'm going to first start with the ordinary thing of disclosing that I have gotten some research support from Novartis and from Abbott Molecular, and I have some equity in a startup vaccine company called Sound Vaccines. So we've been hearing about Gottfried Schmier and his life in coagulation, and he had an eminent career in coagulation, so much so that when you're near retirement, it would probably be enough for most people, but not for Gottfried Schmier. So he did a lot of preparation for his retirement. And you can see this quote from describing how the greatest adventures in his life basically began when he retired. But to prepare for retirement, he went to Tulane University, where he obtained a Master's of Public Health. He spent time in Peru and took the famed Gorgas course in tropical medicine. And then he worked with people in the Altiplano region of Peru. He attained a certificate in tropical medicine from the society I belong to, the American Society of Tropical Medicine and Hygiene. And here you can see Gottfried, the retired aged guy, amongst all these young Peruvians and other international students at the Gorgas course. This is a picture of Gottfried working in the Amazonas region. So he would take medical expeditions up the Rio Negro and Rio Amazonas rivers and spend a great deal of time there, not alone. His wife Elizabeth joined him in these expeditions, and they would often travel in, this is a picture that Gottfried himself took, would travel in these boats, both the big ones and the large ones, up the river to access areas that were very remote. And along the way, they encountered lots of malaria, leishmaniasis, and other infections. And he became a real expert in clinical tropical medicine. And he applied a lot of advanced tools. I was always amazed that he was up to date on the latest things, but he also applied a lot of really practical tools. He tells the story of curing a botfly by simply applying some ointment to the skin, not really knowing it was a botfly, and he had no antibiotics. And when the fly popped out later, the patient came to him and said, "You're the best doctor in the world." And he said, "I know." But inside, he knew that he just got lucky with that one. So, I could tell lots of stories about Dr. Schmier. I inherited the parasitology course that he taught for many years. And at the end of the talk, I'll let him have the last word in regard to that. But right now, he would basically be saying, okay, let's get on with the business of talking about malaria. So when Gottfried was in retirement, this was a very remarkable time for malaria worldwide. What had been millions of deaths a year started to become hundreds of thousands of deaths a year. And most of these cases of malaria are in sub-Saharan Africa, and most of the people who are affected are young children. Those are the people who are most likely to die. This was a time of intense optimism, increased funding, and focus on this disease. We learned the parasite genome. We were able to manipulate the parasite, etc. The last couple of years have given us pause. This is our starting point for today, which is that the World Health Organization report for 2018 has basically shown that that downward progress has, from 2015 on, stalled. And they've in fact identified 10 high-burden countries, 9 of which are in Africa, and then India, where cases of malaria are actually increasing, despite the fact that we have bed nets and insecticides and know a lot more about the parasite than we have before. So why is malaria elimination so difficult? It's really hard to study this on the whole world, and so people tend to try to study malaria eradication in a microcosm, and one such microcosm is Bioko Island, which is off of Equatorial Guinea. This is a demonstration site for malaria elimination, and our own Diana Hergott, who's now a PhD student in epidemiology and is sitting right over there, worked on Bioko Island for part of this program that is still running, but ran from 2004 to 2016, and Diana and her colleagues recently analyzed the data. So I want to show you Bioko Island. So this is us. We're flying northeast in the Gulf of Guinea, and we're flying toward continental Africa. But before you get there, you come across this island that's a little bit bigger than Maui. And on the northern end of Bioko Island sits Malabo. Malabo is the capital city of Equatorial Guinea. The rest of the country is on the mainland. Most of the residents on in Bioko Island, about 300,000 people, they live here in Malabo, and that's where most of the malaria cases occur. So if you look at the data from 2004, where there were places in 2004 where there were more than 1,000 bites per year that were infected per person. And by 2015, that had dropped to 15 bites. Nonetheless, there was still fairly high prevalence malaria in Bioko Island. It went from 43% of people to 8%. The problem is when you zoom out from Bioko Island, you see how massive the landmass that we're talking about where malaria is endemic. This is Democratic Republic of Congo, Uganda, Kenya, and on through Mozambique. And when you zoom out and look at all of Africa, you kind of wonder, how on earth can we use existing tools to eradicate malaria if 12 years of work on Bioko Island has failed to eradicate malaria? The WHO has recently released a plan called High Burden to High Impact, where instead of trying to shrink the map from the outside in, they're going to take these tools to the highest burden countries and apply them there where previously they hadn't really had that focus. But we really think that other kinds of tools are needed, tools that really change the way that we can vaccinate against malaria. And so that's what I'm going to talk about here. So we're going to talk about the work that we do in our lab focused on T-cell vaccines, diagnostics, and clinical trials. This is sort of a survey of what we do. And before we do that, however, so two of these projects, the T-cell work and the diagnostic work, are housed here at the University of Washington. And our vaccine clinical trial work is an interinstitutional effort that I'll briefly tell you about that involves other centers in Seattle beyond just the University of Washington. Before I do that, let's make sure everybody is on the same page with respect to malaria. This is a disease that is transmitted by Anopheline mosquitoes, which are depicted here by Dr. Seuss, who, before he was a children's illustrator, drew a lot of pictures about syphilis and malaria for World War II. And these mosquitoes, these female mosquitoes, they take a blood meal, and when they do, they transmit this single-celled parasite to you, which you'll see in a moment. And this parasite goes to your liver for about a week, and after a week it emerges into the bloodstream where you get all of the clinical symptoms associated with malaria. This is a better way to look at the life cycle. So this is an Anopheline mosquito that is following your carbon dioxide plume, and when she takes a blood meal, which she does to nourish her eggs, she injects anticoagulants and she also injects sporozoites. And these sporozoites, they find their way to your bloodstream, and then they eventually find their way to your liver, where they infect a hepatocyte, as you'll see in a moment. And over the course of about a week, they multiply 30,000-fold. After that week, they release little packets of parasites called merosomes back into the bloodstream. These merosomes probably hit the pulmonary vasculature and break open, releasing the truly infectious parasites, these merozoites. So these merozoites bump into red cells, because after all, there's lots of red cells in the blood, and they enter the red cell and form a vacuole where they will then grow for the next 2 days. They form this classic ring that we learn about in microscopy. Now, this is the time in which you can have symptoms, because the cyclical destruction of your red cells gives rise to fevers, chills, flu-like symptoms, sometimes headache and vomiting. If the disease is left to go long enough, you can develop severe anemia, convulsions, lapse into a coma, and even die. The parasite can hang onto your blood vessel wall and evade removal in the spleen, and then eventually it ruptures and releases more of these infectious parasites, the merozoites. Eventually, the parasite also gets cues to say, it's time to get out of here, and it makes sexual stage parasites, these male and female gametocytes, that carry no pathogenic potential in humans. But if they are taken up along with the blood into a passing mosquito, that's when they can do their thing. So when you go into a mosquito, the temperature drops from 37 Celsius to ambient, and this cues the male gametocyte to exflagellate, forming a sperm-like looking cell that fertilizes the female gametocyte, and then this resulting zygote lives in the midgut wall of the mosquito. Along the equator, the next process takes about a week, here maybe 2 weeks, it's temperature dependent. This is where the sporozoites are born. And once they are born and rupture from the oocyte, they find their way to the salivary glands. And if the mosquito can then survive to bite another person, you get the whole life cycle. So this seems like a very tenuous life cycle, like it shouldn't be as prolific as it is. And yet 1,200 people or more a day die from this infection. Most of these are children who are younger than 5, and most of them live in sub-Saharan Africa. So first I'm going to tell you about the work we're doing to try to develop a very different vaccine than what is currently out there. And so we'll focus on that first. First question, and I've given some talks at Grand Rounds here before on some immunology aspects of vaccines for malaria, immune evasion in malaria, these kinds of topics. The first question is there any precedent for making an anti-parasite vaccine? And the answer is no, there is not. There are only approved vaccines for use in humans for bacteria and viruses. Now, some of you have been reading the news and you've heard about the approval in Europe and use in Africa of a vaccine called RTSS that's being rolled out in Ghana, Kenya, and a few other countries. I'll touch on that in a few minutes. If we look at the life cycle of the malaria parasite, we have at our disposal antibodies early on when the sporozoites invade. But during the liver stage, the preeminent cell that can find and destroy the infected cells are CD8 Tcells. These are the same kind of CD8 Tcells that find and destroy virally infected cells or tumor cells. As the life cycle continues and moves into the red cell stage, red cells, they don't have class 1 or class 2 MHC, and so they really can only be taken out of circulation by antibodies or by mechanical mechanisms in the spleen. We can also induce antibodies against the mosquito stages, which forms a kind of vaccine called transmission-blocking vaccines. But that's really beyond the scope of the talk today. We're going to focus on these CD8 Tcells, because if you can make a vaccine at the liver stage and you can kill all the parasite-infected liver cells, then you can prevent all of the downstream life cycle from happening. So the liver stage is a really ideal target because we have about 6 days to kill the parasite, and we have these T cells that can search and destroy. The problem is that the bar is very high. We have to kill 100% of the cells in the liver or we lose. One infected cell that goes forward to the red cell stage is capable of kicking off the red cell life cycle. And the parasite evolves its protein expression as it goes through the life cycle, such that the antigens and proteins expressed in the red cell are really different than in the sporozoite stage. It's almost as if you have 2 different pathogens. So the thing that I always liken this to is trying to prevent metastases in cancer, trying to get at the first bolus of whatever the infectious or cancer is before it spreads, because our ticking clock has 6 days on it, and that's what we have to kill it. So most vaccines that have targeted the parasite in the liver or before the liver tend to induce antibodies. And that's really because the whole vaccine industry has arisen around vaccines that induce antibodies. These antibodies, if you were going to do this in the liver, are intended to prevent the parasite from getting into the liver in the first place. The problem is that the window of opportunity there is small, minutes to hours, and you need to have your antibodies present at a very high titer in order to make this happen. The RTSS vaccine, which is made by GlaxoSmithKline, requires 4 doses and ultimately has an efficacy of about 39% against cases of malaria. It's never shown a benefit against deaths, and that's probably because it needs to be rolled out on such a big scale. That's not to say that it's not a good tool, but it is not a magic bullet for malaria. The rollout of the RTSS vaccine will not precipitate the eradication of malaria. Instead, I think it's a complementary tool to other tools that are also imperfect. And what we would really like is to make a malaria vaccine, the efficacy of which is much higher, 80, 90% or higher. Now, there are efforts underway to make antibody vaccines better by finding the perfect broadly neutralizing antibodies or improving antibody avidity. But we have focused our efforts on this CD8 kind of mechanism of immunity. And the tool that we have used to understand this are live attenuated sporozoites. So you'll recall when the mosquito bites, she injects sporozoites into you. This is a picture of a sporozoite labeled with antibodies. They're these whisker-like cells that find their way to the liver. So this is the real parasite. And if it is alive, but only goes to the liver and then dies in the liver, you can make and elicit a really nice, broad, complex immune response. And if you repeatedly do this, you can engender good immunity to malaria. This doesn't happen at physiologic exposure levels of the parasite. We give 100,000 or more sporozoites, and they're attenuated. And so a lot of different strategies have arisen. Genetically, this is GAS, genetically attenuated sporozoites, RAS, radiation attenuated sporozoites, and other methods. The problem with this is that even though we can experimentally protect people at high rates, these sporozoite vaccines have to be made in mosquitoes, they must be cryopreserved, and they do not work if you do not inject them intravenously. We basically are mainlining them right to the liver, which is where they need to go, but if they get hung up in the skin, the vaccine potency goes way down and you lose efficacy. So what's really remarkable is that in non-endemic sites, this means the US and Europe, where people have no baseline immunity, these kind of sporozoite vaccines can achieve vaccine efficacy against infection of up to 100%. Later, I'll tell you about what a challenge trial is, but these vaccine efficacy data are not against cases. This is against infection. So when you have 100% vaccine efficacy, that means that everyone in that group who got vaccinated had 0 evidence of the parasite coming out of the liver and causing red cell infection. So that's really tremendous. But we don't just want to protect travelers in the US military, we want to protect people who live in Africa and Asia. And those people have grown up in a region where they've been exposed to malaria again and again. And so they have a complex milieu of antibodies and T cells, adults more so than children. And when you take this vaccine and use it on them, it doesn't work nearly as well. And it is not surprising that that's the case, because the vaccine is not very different from the live parasite. So what we have tried to take away from this and learn from is, how on earth do these things protect when they do so like they did in this study? This was a clinical trial where people got 1, 2, 3, 4 doses of these sporozoites. This is 270,000 sporozoites. And then they were challenged, purposefully challenged with malaria parasites at 41 weeks and also at 80 weeks. 80 weeks is effectively a year, more than a year post-immunization. Of the people who were challenged a year post-immunization, they were all protected. If you look at their antibodies do not explain why they were protected. If you look at their peripheral blood mononuclear cells, like many vaccine studies do, their T cells that are floating around in their blood, there is no clear correlate of protection. So what is responsible for this protection? The whole immunology field in the last 10 or so years has woken up to the idea that we are not a well-mixed mixture of T cells from head to toe, but that we have very tissue-specific immune responses. And this makes good sense. In the case of malaria, the cells that protect us when we use sporozoites, we think are CD8 liver resident memory T cells. They live in the liver sinusoids. They, like other T cells, recognize infected cells in the context of class 1 MHC. They express a cocktail of surface markers that allow us to find them by flow cytometry. They express cytokines that allow them to kill infected cells. And they can be induced by expressing the antigen in the tissue of interest, in this case, the liver. Over the last several years, 3 groups, including ours, have published on what we will call preclinical prime and trap vaccines to try to increase these cells in the liver. First, Bill Heath's group in Melbourne, Australia, our group here at the UW, and Adrian Hill's group at Oxford University, all by different methods. And I'm going to tell you a little bit about Bill Heath's stuff. First, by showing you these cells in real life. So the Heath group took green fluorescent protein labeled T cells that went on to become resident memory cells, and they did intravital imaging. And you can see these cells are patrolling the liver sinusoids at about 10 microns per minute. If you zoom out, you see these cells that are the slow transiting cells. These are the liver resident memory T cells. If you look where there's bloodstream, you see other cells that are zipping through. These are peripheral T cells that are probably coming in on the blood flow and zipping out on the blood flow. It is these resident memory T cells that seem to correlate with protection. And so the question is, how do we make this happen in people? We cannot take out people's livers and try to study their resident memory T cells. So to do this, we start with mice. And the first vaccine that we have worked on, our prime and trap vaccine, goes like this. This was work done in my lab by Taylor Olson, who's now at the Fred Hutch Research Center. So we have used a DNA gene gun to kick off the immune response. I'll tell you about the gene gun in a few minutes. This is to kick off CD8 T cells. And once those CD8 T cells are primed, we then use a single dose of intravenous sporozoites that are injected. They go to the liver where they express their antigen, and the T cells come running. So prime and trap. And the question was, is this more protective than just giving this sporozoite over and over again, which has been done for 40 years? The priming is done using a gene gun to deliver DNA. And the DNA vaccine work in my group has largely been led by Brad Stone, who's here, and in collaboration with Deb Fuller, who's in microbiology, who is actually one of the co-inventors of the gene gun. So the idea with a DNA vaccine is that DNA encodes RNA and RNA encodes protein. And so if you encode an antigen from the parasite and you put it in a host cell where it is expressed, it will be recognized as a foreign protein and an immune response will ensue. At least we hope. There are a number of ways to induce, to deliver DNA for DNA vaccines, and our preferred method is the gene gun. So this is a gene gun. A gene gun is basically a helium pulse that shoots tiny 1 micron gold beads coated with the DNA of interest into the skin. The skin is full of lots of different cells, including dendritic cells. And some of these 1 micron gold beads end up inside of dendritic cells, other cells as well, where the DNA in plasmid form is expressed, the antigen is recognized as foreign, and the immune response is kicked off. The gene gun is very dose sparing. It is painless. It is loud, but it is painless. And it is very good at priming T cells. So at first we thought, we're going to use the gene gun to save the whole world. But this slide will tell you why that won't work. So when we took our animals, we can immunize them. This is GG, gene gun, and then CSP here is one of our antigens of interest. This is a well-known protective antigen in experimental settings. When we use the gene gun, we can take wild-type BALB/c mice, immunize them, and attain 25% of the CD8 T cells specific for that one antigen. So none of us walk around with anywhere near that frequency devoted to a single antigen, but this is an experimental system. And at the peak of that response, if we challenge those animals, we get sterile protection in the majority of animals. That is not true for other antigens like these, the names really don't matter, but other antigens that are equally immunogenic but not protective. One of the real difficulties in malaria is separating the wheat from the chaff. There are 5,200 genes. Many of these induce immune responses. Most of those immune responses are not protective. CSP is special. If we let the CSP immune response settle down to, let's say, a month post-immunization, now the frequency of those T cells is down to 4%. And now if we challenge mice, we completely lose this protection. So this is T cells in the blood. This doesn't tell us anything about T cells in the liver. Is there anything about a gene gun and DNA that would say, hey, go to the liver? Why not go to the liver? There's nothing that would say that. So just let me pause for a second. We asked the question, is there something wrong with DNA vaccines that lead them to not go to the liver? And so we took DNA, we actually delivered it to the liver of the mice, and lo and behold, when the DNA is expressed in the liver, the T cells come running, and they form the right kind of immune response. So we said, okay, well, we can't do that method, which is called hydrodynamic transfection. We can't do that on people for reasons I'll tell you later if you're interested. We need a much more elegant way to do this. So we asked, well, could we use sporozoites as a surrogate for this? After all, they're professionals at going to the liver. If we do what most people do, which is look at the spleen or blood, these are like immunologists' favorite organs, and we either gene gun immunize or give irradiated parasites one time, what we see is that the gene gun is really good at inducing strong T cell responses in the spleen. Same is true in the blood, but it's not shown. If we do a prime and trap vaccine, DNA followed by sporozoites, we see the same thing. What is different is if we look in the liver. If we look in the liver at resident memory T cells, it is only the prime and trap vaccine that rises above all the rest as the highest frequency resident memory T cells. And this is the only regimen that basically results in 90 to 100% protection in all of the situations that we have tried in BALB/c mice. And so we have now gone on and looked even later post-immunization and have data that 90 to 100% of animals are protected up to 4 months post-immunization when we give DNA followed by a single dose of irradiated parasites, which is much easier to do than 4 or 5 doses of irradiated parasites. We're evaluating a lot of different parameters around this, and we are looking for other antigens. We're also looking for ways to replace the sporozoites with something that doesn't have to be grown in mosquitoes, injected IV, cryopreserved, etc. But for now, we are also moving this forward, and we are moving this model into non-human primates. The reason to do this is that many vaccines have failed when they make the jump from mouse to non-human primates and humans. And what non-human primates allow us to do is, first of all, we will be looking in the liver using a molecular endpoint to say how many malaria parasites in immunized versus placebo control animals are in the liver. And the other thing that having access to liver lets us do is it lets us say, did the cells that we think are mechanistically responsible for protection, did they accumulate in the liver like they did in the mice? If they do in the monkey, then hopefully that would be true in the humans, but we cannot take wedge resection biopsies of human livers. And so the non-human primate work is moving forward at a rapid pace, and that's going to be the work of Melanie Shears, Chaitra Parthiban, and Jokichi Matsubara, who are here in the front row. And so that will start this summer. So the next steps for our vaccine work are to refine this prime and trap vaccine using the mouse and non-human primate models. We would really like to get away from sporozoites, and so we're interested, we've been trying different kind of experimental tools for the trap. We're also looking to identify novel protective antigens. That's what Irene and Brad are working on. And importantly, we cannot have a vaccine that only works in people who have never been exposed to malaria. And so what we're really trying to do is think about this problem that has already been witnessed, which is that some vaccines work well in the US and Europe and don't work well in Africa. We need to come up with a mechanism that can overcome that, not be crippled by that. So now I'm going to briefly talk about our clinical trials, and the main reason I'm going to do that is so that you can understand how the diagnostic test that we have developed is being used in those trials. I have previously talked about the Seattle Malaria Clinical Trials Center here at Grand Rounds, and this is an interinstitutional effort combining Seattle Children's Research Institute, the Fred Hutch, and the UW. It is led by Jim Kublin, who is at the Fred Hutch, and is also the director of the HVTN. So what we do is leverage the real clinical trial expertise and sample processing, research sample processing excellence at the Hutch. We use Seattle Children's Research Institute. This is the building that used to be the Center for Infectious Disease Research to make GMP-grade mosquitoes for our human challenge work. And then we use our diagnostic lab here for both safety labs and PCR testing, and then we provide physician support. So what is controlled human malaria infection briefly? This is a model that in some ways is the envy of a lot of other infectious disease systems where you can randomize people to drug or no drug, vaccine or no vaccine, and then we can actually infect them on purpose with either mosquito bites or with intravenous administration of the parasites. And we can then follow them very closely over the following month to see, did the vaccine work? If it did not work, the patients are treated quickly and have really no significant adverse events. Over the past years, we have done an increasing number of mosquito bite or intravenous administrations of parasites. Every single vertical line here is a day that we gave people parasites from 2010 until 2019. And this has led to all of these completed studies, some of which are published, some of which are in various stages of being published. I'm not going to talk about these individual trials. We collaborate with a number of people, Stefan Kappa, Lisa Jackson, Janine Menza. Some of you may know some of these people. These are really complicated trials. The choreography of them requires lots of people to come together really on one day to allow the trial to go forward. And this is different than a lot of clinical trials where you enroll people in clinic on a sort of rolling basis. So I thought I would point out some of the differences. We, like all trials, have really strict inclusion and exclusion criteria. We have to have really responsible, trustworthy people who are going to come to clinic every day after we infect them on purpose with Plasmodium falciparum, because if they don't, they could get really sick. And all of the participants are usually enrolled on the same day. We achieve safety in these studies by using a drug-sensitive strain, by doing really high analytically sensitive testing, which I'll tell you about in a moment, and by following up these people really closely. We do lots and lots of lab testing. So in the Novartis-sponsored CAF156 study, this department, lab medicine, not for PCR, but for just safety labs, produced 29,531 endpoints that went into the database. The choreography is, as I said, really critical, especially in mosquito bite studies, because you can't just put them on the shelf and they'll be good in a month. They are good for a few days and that's it. And these are basically like FDA vetted GMP little flying syringes that have to be within very close spec. So one year, the mosquitoes we had here were not going to be eligible to be used. And there is only one other place in the country that is okay for us to obtain mosquitoes from. It is a lot of work to get these trials to all come together, to get people to take time off of work, the participants, to get all the clinics lined up. So rather than cancel the trial, myself and two other people flew to Rockville, Maryland, and in 43 hours we drove a very expensive case of mosquitoes back across the country, and we did the trial. So it was fun as well, and it worked. Otherwise I wouldn't tell you this. But sometimes we have to go to really great lengths to make these trials happen, either to get the trial started or to follow up on participants. One of the things that has made these trials easier is the molecular diagnostics that we do here at the University of Washington in our lab that we call the Malaria Molecular Diagnostic Laboratory. The gold standard test for malaria for more than 100 years has been repeated blood smears. And blood smears are a great way to diagnose malaria in travelers, people who are sick and febrile, but we don't really want our participants to be throwing up and be sick and febrile. And at the very limit of detection of blood smears, they're not particularly quantitative. So it's been known for a long time that we could use molecular biomarkers to identify the parasites. And the biomarker that we use in our lab is the 18S ribosomal RNA. So we actually study the RNA itself. Many other labs study the ribosomal DNA, that is the genes that encode these RNAs. The reason that we study the RNA and do reverse transcription PCR is that the parasite does part of the work by already making thousands of copies of the RNA in every single cell. So every single cell has 3,500 to 10,000 copies, which means we can detect a single parasite in a pretty big sample of blood. And this is just to show the difference between doing with and without reverse transcription by real-time PCR. We get a very accelerated detection. The test that is in its third generation and is run at Harborview has 3 targets. We test for P-falciparum 18S ribosomal RNA, we test for a pan-Plasmodium 18S ribosomal RNA, and we have an endogenous internal control. In addition to using this for clinical trials, we've also brought this on as a lab-developed test, so it is clinically available, though it is not intended to replace blood smears as a first-line diagnosis of malaria. It's really intended to address unusual cases that have perhaps evaded that, or to be applied in some of our other clinical trials that involve people here in Seattle outside of the CHMI model. Because our assay was run in a clinical lab and because lab medicine has such a good reputation, we were selected to basically be an emissary of the malaria field, to go to the FDA and try to replace blood smears with something else, or at least allow something else to be permitted as an endpoint in phase 2 and phase 3 clinical trials where the data might be used to register drugs and vaccines for licensure. What do you do when you're an academic lab? We're not trying to sell a test in a box. So the avenue that you go down is what's called biomarker qualification. Biomarker qualification is more than just analytical validation of the test. It's separate from simple clinical validation. And so let me just tell you about it. So what we do is analytically validate the test. We say, we detect the 18S ribosomal RNA, and our test, when we say it's positive, that's what it's detecting. These are the interferences. This is the limit of detection. Everybody in this, most of the people in this room are well aware about analytical validation of a test. Clinical validation of a test is, when this is positive, the person either has or will go on to have the disease that we know as malaria. In this case, we then overlay a context of use. And the context of use here is that we intend to use this in human challenge trials. We actually don't want them to get the symptoms of malaria. And so our context of use is, we're going to use this test in these clinical trials. When it's positive, it's going to induce treatment. And when it's positive, that means the person was infected. You can then decide what that means for your vaccine or drug. We are not the only people who have known for a long time that molecular testing is more sensitive and therefore allows for accelerated detection compared to blood smears. This is data from a paper where Mariko Sile and Ming Cheng are the co-first authors on, where they looked at our data and data from a number of other clinical trials. And what you see is each row here is a different cohort of a different clinical trial, where the boxes are nucleic acid tests and the circles are blood smears. And in every case, the boxes are to the left of blood smears, because in every case, molecular testing is accelerated because it is more sensitive. This is not surprising, but it is sort of beautifully illustrated here. It's not just blood smears that we beat. We also beat symptoms when we use molecular testing. This is data that we submitted to the FDA showing that when our molecular test is positive, it is usually positive 2 days or so before any symptom. Any symptom in these clinical trials can even be like, I'm tired today because I've come into this clinical trial center every day for 14 days and I'm sick of it. These numbers get even bigger if we look at grade 2 adverse events. We accelerate the detection against the events we really would like to prevent even more. As you ratchet up the number of parasites where you would call your molecular test positive, you lose that sensitivity. And so our test sensitivity is down here in the most sensitive range. The idea with this, of course, is that let's say this is a patient's parasite density rather, over the course of their infection in a study where they were tested by blood smears. Now, blood smear is applied when every time, but it's only positive here, and when it's positive here, they initiate treatment. When we use a biomarker, we call this a biomarker, we can detect the infection confidently and earlier, and we can induce treatment earlier in people who are not protected by the vaccine or the drug, or who happen to be in the placebo control group, because there's always a group who's going to get malaria. And when we do that, we treat them earlier, we have fewer adverse events, we can have eliminated what we call the hotel phase. So we used to put people up in a hotel from about day 6 to about day 21, where they would get blood smears in the morning, in the evening, and on demand for any symptoms. And you can imagine this is really burdensome. The first few days are great fun, and then after that it's terrible. So no hotel phase. We have happier clinical trial participants, more quantitative data, cleaner endpoints. All of the data we submitted to the FDA was using the sort of retrospectoscope. Completed trials. If we had done this, we would have gotten a better outcome. We then did the DSM-265 study, which was a drug study in collaboration with MMV, where we found that when we used a molecular diagnostic endpoint, we did indeed detect infections earlier, we treated them earlier, and the patients consequently had fewer symptoms. And just to share a little bit of specific data, we did a cohort that was identically performed in Germany, except that in Germany they used a blood smear endpoint, and here we used our molecular diagnostic endpoint. And whereas in Germany they had, and same number of participants, et cetera, they had 60 adverse events, including a number of grade 3 adverse event fevers. We had 22 adverse events and no grade 3 events. So we have now prospective data that this is a good way to do these trials. Safer, more quantitative, et cetera. So after 4 years, in October of 2018, we received FDA's agency-wide qualification of this biomarker. And this means that the biomarker can be used not just in the drug part of FDA at CDER, it can also be used to support vaccine studies in the vaccine and biologics side at CBER. This was a huge submission supported by this department. We had 58 appendices and over 1,000 pages of information that went to the FDA, got to participate in face-to-face meetings with the FDA, all to get this 1.5-page letter that said, you're good to go. They actually were very collaborative, and we are going to continue to work with them to expand the context of use outside of non-endemic areas to apply to doing these same studies in endemic areas, where you can imagine the situation is a little bit more complicated because you could have a number of different Plasmodium parasites. What else are we using the biomarker for? So one of the problems that we face in malaria outside of clinical trials is what a lot of people have referred to as the iceberg problem, which is that the people who are above the waterline and are symptomatic and febrile and clearly blood smear positive, this is the minority of people. Most people who have malaria infections are below the waterline. And in malaria, nobody really knows how deep the iceberg goes, nor do we know how sensitive or how far down we need to see in order to guide elimination effectively. So the testing that detects people in this symptomatic range is understandably less analytically sensitive, blood smears and rapid diagnostic tests. When you get to molecular tests, they can be much more sensitive, hundreds of times more sensitive than these other methods. And the question is, what is the trade-off between a really highly sensitive but perhaps more expensive test that acts more like a crystal ball to tell you the whole truth, and doing a much less expensive test at the patient bedside where you could do more of them and saturate that testing? I don't have an answer for this, but we are working on a number of studies. We're trying to lead one study to understand the asymptomatic natural history of malaria infection, and we work with a number of other collaborators to try to understand this problem. I thought I would also tell you about this case that I think is, hold on, I'm all caught up here, that I think is illustrative of a different problem, but one that we actually sometimes we haven't faced this case in Seattle, but we do have bone marrow transplant patient population here. So a few years ago, I got a call from the NIH Clinical Center, where we have done some collaborations, to say, would you mind doing some research testing on some samples from a participant who was in a clinical trial, and this was a participant story. This was a 32-year-old man who had come from Nigeria. He had sickle cell anemia. And what's remarkable now is that people are doing bone marrow transplants for sickle cell anemia. And I think there was a 60 Minutes a month or two ago talking about the improved outcomes with sickle cell anemia by increasing the conditioning regimens. Anyway, this man came from Nigeria, and he reported, as many people from Nigeria would, a history of malaria episodes. But he was tested by the NIH's PCR, which has a limit of detection of about 500 parasites per milliliter, more sensitive than blood smears, more sensitive than rapid diagnostic tests. He was negative. And so then he was suppressed, and lo and behold, he went on to develop malaria infection. And because he had sickle cell anemia, he had auto-infarcted his spleen, which is your malaria filter. And so his malaria filter wasn't working. So in addition to having this undiagnosed infection that manifests during bone marrow transplant, he had a really extended course of clearance because the organ intended to clear it wasn't working. When we looked back at his pre-transplant sample, it was clearly positive by our tests. And so we have been working with the NIH and talking to some other people about how many people are doing this. Lots of people have sickle cell anemia, and this may be an area that we could help out with. It's a niche area, but transplants for sickle cell anemia would be a great change in that quality of life if they were to work. Okay, yep, there's our data point. So, so I just told you a few things that the MMDL has done, the items in red. We've participated in a lot of other really cool collaborations because the test allows us to give insight to a lot of different kinds of studies. This is the work of Mariko, Curtis, and Ming, who are up here, and also Nahum Smith, who has recently departed our lab, but I see is sitting in the back corner. Thank you for coming. We have done over 13,000 samples in this lab. This lab is at Harborview, but next year we'll be moving to South Lake Union so we can all work on one footprint. So I'm going to summarize here, and then I have just a couple more slides. I told you about prime and trap vaccines and how they might lead to more effective outcomes based on CD8 Tcells. I told you about our clinical trial center here in Seattle, which is one of few centers in the world that can do these kind of mosquito bite human challenge studies. And then I told you about our RT-PCR testing approach that has gained us a lot of collaborations, an FDA biomarker qualification, and led to a lot of really good science. So I have a lot of people to thank. I first want to say I get to work with a really great team who are all here in the second row, and one of whom's over there. And I really love working with you guys, and you sort of bring it every day, and it's really great to see. We have a lot of other collaborators, and Brian Welt as well, who's here in the grants team. I once tried to enumerate how many emails go between me and the grants team, but there's no filter to figure that out. It is maybe a million. And I'd also like to thank Jim Kublin, who is actually the person who clued me into the fact that we needed a molecular diagnostic test when I was first a resident here. And I said, oh, we're in lab medicine. We do great testing. We'll do it. And I sort of bluffed, and it has turned out really nicely. So in closing, I want to have Godfrid have the last slide. I agree with all of the things that you said before. He was all of these things to me. He was all of these things to a lot of people in this room. This is a picture of him teaching in clinical microbiology rounds, and he would very faithfully come once a week with slides that were prepared, with glass slides that he had circled the place so he wouldn't waste your time, so he could go right and find that rare parasite that was on that slide. So he was a really dear friend. And I want to show you this, some video here. So first thing is that he used to dedicate each lecture, each series to his wife, Elizabeth, who I learned passed away not too long ago. And he would often use this slide to talk about joy. This was a picture of kids jumping into the Amazon River.
We have lost some say how to get out of that perspective. Because I think I get when my patients have to learn. So with these wise words, I close.
All right. Thank you for listening.
Questions, please raise your hand. We have a microphone.
So lovely presentation, Sean.
Thank you.
In a lot of cases where people have chronic or recurrent infections in endemic areas, they develop regulatory immune responses to control their cellular immunity because of the pathogenesis of immunopathogenesis to balance that with parasite or other microbe eradication. So maybe those are interfering with your vaccine responses in endemic areas. Is there a way to transiently, reversibly inhibit those regulatory responses to adjuvantize your vaccines in those areas? I think there's work of that nature going on.
Yeah, so the question for the microphone was that people who have repeated chronic malaria infections have regulatory immune responses that may serve to reduce the immunogenicity of the desirable responses, but also serve to temper the pathogenic responses. So it's absolutely true. There's Tregs, there's exhausted T cells, there's all kinds of stuff. There is some work on mice to show that if you relieve the T cell exhaustion with antibodies, you can precipitate clearance in a much cleaner way. But it is, of course, a balancing act. And for us, I think what we're... So you definitely don't want to vaccinate in the setting of an ongoing red cell stage malaria infection. And a lot of groups will treat and clear people with drugs, but monitoring that throughout a multi-month-long study is really hard. We can't cohort people for 3 months just so they don't get malaria on their way home from the clinic. There are ways to relieve immune exhaustion with antibody infusions and other kinds of things, but I think mostly we've thought about those in experimental medicine settings as opposed to ways forward in the malaria field. I kind of see that a lot of public health interventions are heading us in a direction where there is less malaria. And so one simple-minded way to do this would be to immunize people in the low season, where maybe you don't have to do anything, but that it's an opportune time. Now is the time, so when they're not focused on their ongoing infections. It's a great question and area of a lot of research, not ours, but we'll see what happens.
Hey, Sean, that was a great talk. Thanks very much. In the successful mouse studies with your prime and trap setup, is there any evidence of hepatitis?
It's a good question. So you would think that if you started a rip-roaring immune response, it's not actually that rip-roaring. The liver is a big place, and if we put in a couple 100,000 parasites, it's still very small. The best evidence I can give you is that the groups that are doing these studies in humans are acutely aware of the need to not cause hepatitis. And when they give hundreds of thousands or even millions of sporozoites, they monitor ALT, and that doesn't really go anywhere. So we have come up with some experimental methods like hydrodynamic transfection, where we would essentially be transfecting all the cells in the liver, and we did not see the ALT. I'm looking at Melanie to double check. We did not really see the ALT meter move there either, but we didn't check them every day for a long time. Mice only have so much blood.
Sean, a beautiful talk, and I know if Gottfried were here, he would have loved to hear it.
Thanks.
My question is related to the adjuvant effect of DNA itself, and some forms of DNA can serve as an adjuvant. Is the DNA that you're using have that effect, do you know? Or if not, could you use high CpG DNA that does have an adjuvant effect when you
So
give it the immunization?
The question, and sorry I didn't repeat your question, the question was, is the DNA self-adjuvanting? Could we do things to make it more adjuvanted? So you can use CpG nucleotides to make it more immunostimulatory. We have played around with that, but we don't do that currently. Instead, what we do is we'll have a plasmid or plasmids that encode one or more antigens, and we'll have another plasmid that encodes, in this case, heat labile enterotoxin. It's called LT. It's from E. coli. And it is then co-expressed in wherever those cells end up. So it is an adjuvant, but it is a very local adjuvant intended to help provoke the response only in the transfected cells, because we wouldn't want to give it systemically. So that's our approach. The problem with immunology experiments in general is that there are a million ways to do them, and you have to pick one and go. Great. Thank you, everybody.